Okay.
All right, we are now recording. Thank you very much, John, for agreeing to give a little seminar. And go ahead.
All right. Well, yeah, I'd like to begin by thanking Kate for the invitation to speak here today. I wish I could be there in person to meet you all. But for those of you who don't know me, my name is John Chaput, and I'm a professor of pharmaceutical sciences at the University of California, Irvine. My sort of research focuses primarily on the engineering of polymerases that allow us to synthesize, replicate, and evolve artificial genetic polymers. And so today what I thought I would do is kind of talk about sort of our work in the area of redesigning the genetic polymers of life. Just kind of by way of background a little bit, we realized many years ago that a lot of the major technological advances that have been made in biotechnology and biomedicine have largely been advances that were based on either trying to control or understand the flow of genetic information in biology. And, we wondered or asked the question at the time, what if we weren't constrained by the natural genetic polymers? What if we weren't constrained by DNA? What new sort of discoveries could we make, or what new kind of technologies could we develop? And so we began in the early days kind of writing schemes like this, kind of showing the exchange of genetic information from DNA into what we called XNA at the time, which was really just sort of a generic kind of placeholder for any type of genetic polymer that might have a different sugar phosphate backbone than DNA, and kind of looking at this reciprocal exchange of information where you could think of it sort of in the synthetic biology world of kind of coding and decoding information. But you could also think of it in kind of a biological context of transcription and reverse transcription. And the idea or the vision at the time was that if we could sort of develop a system like this, then perhaps one day we could get to a point where we could replicate XNA independent of DNA. And then if we could even do that, then we might be able to go one step further, which would be to see if we could translate XNA into proteins. Now, of course, these are all like, hugely challenging projects. And so really what we've done over the last 10 or so years is kind of focused on this part of the equation, which is kind of highlighted in this red box here, which once again is this kind of exchange of information between DNA polymers and XNA polymers. And we realized that if we could do this and we could do it very efficiently, that it would open, that just this equation itself would open up lots of sort of new opportunities. And these would be opportunities in areas like information storage or new therapeutics, or perhaps even new diagnostics. And so although we've worked on a lot of different sort of artificial genetic polymers over the years, my lab has largely focused on a particular type of system referred to as Threose nucleic acid, or TNA. So TNA has the same 4 genetic letters as DNA, kind of shown here on the right, so the ATGC genetic letters, but it has a very different sugar phosphate backbone. So TNA is composed of these repeating units of 3O-furanylyl nucleic acid. So it's a 3O sugar, a 4-carbon sugar, that's then linked together by these somewhat unusual 2', 3' phosphodiester linkages. Now, we like TNA for a couple of reasons, and the first of which is that TNA is sort of unusual among a lot of the different XNAs that exist, in that it's able to base pair very efficiently with DNA as well as with RNA. And so a DNA TNA duplex, a chimeric duplex, would have a TM that's very sort of comparable to, say, a DNA RNA duplex. And so what we're showing on this slide here is a crystal structure of a TNA DNA duplex, in which one strand, the one on the right in purple, is entirely composed of TNA, and the one in green is composed entirely of DNA. And so you see a very sort of natural looking helix when you look at it. And if you kind of go in closer detail, you see the very nice formation of these Watson-Crick base pairs, again, between TNA and DNA. And so this property is important because it allows for the transfer of genetic information from one type of genetic polymer to another, but it also allows us to take advantage of all the technologies that have been developed for DNA. So this would be all the technologies that have been developed for DNA synthesis as well as all the technologies that have been developed for DNA sequencing. Okay. So the other property of TNA that we really like as well is that TNA is 100% biologically stable. Okay. So we have yet to find a nuclease or an enzyme in biology that will degrade TNA. And this is really just kind of an example. It's a little bit older data at this time, but I think it's very, very telling. So what we're looking at here is two different TNA oligonucleotide sequences that were prepared by solid phase oligonucleotide synthesis. And what they're doing here is we're incubating them under sort of the harshest conditions that you would use sort of normally to evaluate, say, a therapeutic oligonucleotide. So that would include 50% human serum in medium, as well as, say, concentrated human liver microsomes. And what you see is after 7 days at 37 degrees, we see no degradation. And that contrasts quite significantly with sort of standard chemical modifications that have been studied over the years. So just one example would be like 2' fluoroRNA. So this is an RNA analog where you have a fluorine at the 2' position, as well as its Aptamer. So the fluorine's in the upward configuration here. And that's referred to as fluoroarabinonucleic acid. So these are sort of two sort of examples of nucleic acids that have been well studied. And in these assays, what you see is these examples, these particular analogs have half-lives of, about 45 minutes in concentrated human liver microsomes. And they also show similar degradation profiles in human serum. So TNA is substantially stable than most of the analogs that are available. Now, if you want to sort of be able to work with TNA, in most molecular biology applications, ultimately you're going to have to find a way to copy DNA information into TNA, and then eventually copy that information back into DNA. And so again, we can think about this as a problem involving transcription and reverse transcription. And so, nearly 20 years ago, we started looking at this problem. What we learned is that natural polymerases are not particularly good at recognizing TNA substrates. And it doesn't really depend on really makes no difference. So whether TNA is, say, a triphosphate or a template, okay? And so essentially what the polymerases will do is they'll recognize, say, a DNA primer template duplex. They might add a couple of nucleotides, and then they just pause or chain terminate. And so this really kind of brought up two pretty significant challenges at the time. And one challenge is really just the substrate problem. So TNA is not a common analog. TNA triphosphates are not commercially available. And so if we were really going to work in this space, we'd have to develop the synthetic protocols for producing these molecules. And not only would we need to make them, but we would need to make them on the scales that were large enough that we could have enough material to work with. And so we spent a lot of time doing nucleic acid chemistry and developing new protocols for not only synthesizing TNA triphosphates, but just synthesizing triphosphates in general on a large scale. And unfortunately, I don't have time today to talk about the chemistry of that problem, but instead I want to focus on this other problem here on the right, which is also a problem, probably even a bigger problem, which is this polymerase problem, right? At the end of the day, we need to invent or create our own polymerases that could synthesize TNA. And so this is what you're looking at here is a crystal structure. You're kind of focusing on the active side of that crystal structure. And this is one of our first generation TNA polymerases that was created in the laboratory. And what you have here on the left in yellow is this is the DNA template. Then you have the DNA primer, which terminates in a single TNA nucleotide. And then you have a TNA triphosphate that's bound in the active site in the closed conformation, which is then poised to form the chemical bond. And so essentially we had to create polymerases like this, and we had to make them or evolve them to the point that they would be very efficient. Okay, so how do we do all this? Well, in my lab, we've tried a couple of different strategies, but at the end of the day, we really have focused in on using microfluidics to evolve these types of enzymes. So we evolve our polymerases in water oil droplets, okay? And the way this works is you create a polymerase library at the DNA level. You transfect that library into E. coli. You then grow and express your E. coli so that each E. coli is essentially expressing a different polymerase variant. You then encapsulate that E. coli in a water oil droplet created using microfluidics. You then lyse the E. coli cell. This will release the plasmid, and the enzyme into the surrounding solution. And so now you have this genotype-phenotype relationship such that if the polymerase is active and it performs the task you want, so let's say it synthesizes TNA, you can recover this droplet and you could determine the sequence of that polymerase by sequencing the plasmid. And so this is sort of a very, ultra high throughput kind of strategy for evolving these polymerases. It's also has, really, I think, favorable economies of scales, especially when you're working on triphosphates that you have to prepare yourself in the laboratory. So overall, this is really a two-step microfluidic process, and we call this process DROPS, which is an acronym that stands for Droplet-based Optical Polymerase Sorting. And so we have sort of one microfluidic device that allows us to make our initial water-oil emulsions. So we have a population of these emulsions that we create. Inside the emulsion, you have your E. coli, but you also have your substrate, okay? And then we collect all of these droplets in a tube. We work with thermophilic polymerases. So essentially what we do is we heat the population of droplets up. These droplets are very stable, but the E. coli at a certain temperature will burst and release their polymerase and their plasmid into the surrounding solution. We then incubate it for a defined amount of time. And after that point, we then sort them in a separate microfluidic device where we flow the cells individually through the microfluidic device, where if they're a fluorescent cell, they then get recognized and then get sorted and collected, and the non-fluorescent droplets go to waste, okay? And so this is all based on fluorescence detection. So just to give you an idea of what this looks like, this is a movie kind of showing droplet formation. So what we have here on the left is our microfluidic device on an inverted microscope that's connected to a high-speed camera. And as we sort of merge these fluids together in this microfluidic device, we get these very nice, uniform, well-defined microfluidic droplets. Okay, and so with this device, we're producing droplets at a really fast rate. So it's about 35 kilohertz, or more than 100 million droplets per hour. So we typically would just run a device like this for just a couple of hours, generate a couple 100 million droplets that we could then work with, okay? And in this system, we have an occupancy level of about 10%. And so that's designed so that on average, if it is an occupied droplet, that droplet will only have one E. coli cell in it. Okay, so at the next step, we then would assay them in a functional assay. And so this is essentially a single cell kind of directed evolution approach where we use a very simple sensor. So all the sensors we use in the laboratory are based on kind of this disrupting a fluorophore quencher pair. So in this case, you have a template that has a fluorophore on it. You then have a quencher that's attached to a short DNA primer that's kind of transiently hybridized. Because we work with thermophilic polymerases, we can heat up the system. This DNA quencher will dissociate, and if the polymerase is able to extend to the end of the template, it'll prevent the quencher from reannealing, and you get a fluorescent signal. So it's a pretty simple design, but it's very effective, and you can see that very nicely if you look at this sort of fluorescence and bright field microscope images shown here. And so essentially what we're looking at are droplets that have individual E. coli cells in them. So that's what these red arrows are pointing to. And these E. coli cells have now been lysed, and they've delivered either an engineered polymerase in the top row or a wild type polymerase in the bottom row to the surrounding droplet solution. And what you see, of course, is that if you're delivering an engineered polymerase, you then get fluorescent droplets, where if you're delivering a wild type polymerase, it remains dim. Okay. So at the next point, we then have to go ahead and sort this. So this is our kind of a schematic view of our fluorescent activated droplet sorting device. And again, the droplets are coming down this channel individually. And essentially what they're going to do is pass through a laser right about at this point. And if it is a fluorescent droplet, you will get an excitation emission event that the computer will then recognize. And this is a saltwater electrode that'll fire a series of pulses that change the polarity of the droplet just enough that allow it to hit this divider and kind of roll in. Okay, but if it's not fluorescent, it'll flow along, and there's a second channel of oil kind of pushing it up that forces the non-fluorescent droplets just to go to waste. And that's essentially what you're seeing in this video here. So in this video, just to orient you, the empty droplets are actually dark. They were prepared with a dye in them so that they just appear dark, and the fluorescent droplets are clear. And so all these dark droplets are kind of passing through and going to waste, but every once in a while, you see a fluorescent droplet, which is clear, and it passes through, and you see it gets tugged on just kind of a little bit right here. And that's just enough to allow it to hit the divider and roll in. And so this is a very stable system. We can run these for hours and sometimes even days. And they run at a flow rate of about 2,000 kilohertz, so about 2,000 droplets per second. So a little bit slower than how we make them, but still very, very fast. Okay, so this is just kind of some data on a mock selection, if you will. So what we have here in these images on the bottom are a mixed population of droplets where we have a mixture of both fluorescent droplets and dark droplets. And so in this case, this mixed population is about 25% fluorescent droplets before sorting. And then after sorting, what you see is the positively sorted droplets are more than 99% fluorescent, and the negatively sorted droplets are less than 1% fluorescent. So it's a very efficient process. And then up above is what the software looks like. So this is home-built software, and when the instrument's flowing, these peaks are going by at a tremendous rate. But if you just kind of capture it at one instance, what you're looking at is these little peaks here are the individual droplets, and a large peak would be a fluorescent droplet. Okay, and if that fluorescent droplet exceeds a certain user-defined threshold, we then the instrument will then fire a series of pulses. So that's these three blue lines that allow that droplet to change direction and get sorted. Okay, and so this is just kind of a histogram looking at kind of a droplet sorting experiment. And again, since these droplets are about 10% occupancy, if you would see about 10% of the peaks down here, you would expect that just about all the droplets are getting collected. And so this was just done using droplets that were only expressing an engineered polymerase. Okay, so this approach actually works quite well. And so we've used it to evolve, and we're continually using it to evolve engineered TNA polymerases, because we always want kind of faster, better, more efficient polymerases. But what I'm going to show you here is kind of a movie that's based on 5 X-ray crystal structures that we solved in the lab for one of our, this is our first generation TNA polymerase. Now, these polymerases derive from archaeal B family polymerases. So they have this kind of overall kind of donut shaped topology, but they still have the canonical human right hand as kind of a catalytic domain. And so what you see here is the thumb subdomain on the right. Down below, you have the palm, and then these 2 yellow helices going back are the fingers. You then have the N-terminal, or you have the exonuclease domain, and then the N-terminal domain up here. Okay, and so what I'll show you is kind of walk you through the movie and pause it in certain places just to kind of point out a few things. So that's the apo structure. This is the backside, and you see the helices very well in yellow, and the thumb. And now what's going to happen is the thumb is going to bind the primer template duplex. And so it's going to undergo a major conformational change. And so this is what's happening now. It's binding to the primer template duplex. We're going to just rotate it so we can see the active site a little bit better. And so now this is the binary structure, and we have the TNA triphosphate that enters that structure. And this is the open kind of ternary conformation, as they say. And so what's going to happen is these fingers in orange are going to close over that triphosphate. And this is what you're seeing now. And this essentially is forming the catalytically active complex. So at this point, the enzyme would form a covalent bond between the end of the primer and the incoming TNA triphosphate right there. And once that happens, the enzyme will, the fingers will open up, and the enzyme will translocate to the next position on the template, and the whole process kind of starts over again. So this is effectively a movie showing how an engineered polymerase is synthesizing a completely non-natural or artificial genetic polymer. Okay, so we've, I think the current version of the polymerase that's been published is referred to as our 3rd generation polymerase. This is a polymerase that we refer to as Kod-RSGA. So Kod is the parent archaeal polymerase, so Thermococcus katacarensis. And the RSGA are the gain of function mutations that were identified by directed evolution. So we have 2 mutations in this version that are in the finger subdomain, which again is very important in closing and recognizing the incoming TNA triphosphate. And then we have 2 that are in the thumb subdomain. So one is located in the active site. The other one's very far out on the edge, and it could be involved in dynamics. But in general, the palm is involved in kind of recognizing the primer template duplex and putting it in the right geometry for nucleotide addition. This polymerase has 2 additional mutations we're not showing that are in the exonuclease domain that prevent or silence the exonuclease domain, so preventing it from removing a TNA triphosphate once it's been incorporated. And so that's kind of our current or latest published polymerase. And so we've actually now got a fully replicated system. We can copy DNA into TNA. We also have another polymerase that allows us to copy TNA back into DNA. And so we refer to that as a TNA reverse transcriptase. Unfortunately, I don't have time to talk about that one today, but it does exist, and we can do these full replication cycles. Okay, so what I thought I would do, kind of given the time, is spend a little bit of time talking about, how can a replicating XNA system be used in practical applications. And so there are obviously a lot of different things that researchers might be interested in doing, but one of the perhaps most obvious would be to develop nucleic acid sequences that are totally non-natural, that can fold themselves into a shape that can bind to a target. And of course, we refer to these as aptamers, okay? So most aptamers are created in the laboratory, are composed of DNA and RNA, but we'd like to create aptamers in these sorts of shapes that are composed of entirely non-natural forms of genetic polymers. Now, just by a little bit of background, what are aptamers? Well, this is perhaps the most famous aptamer sequence ever invented. This turns out to be an RNA sequence that folds itself into a shape that can bind to the small molecule target ATP. And ATP is, of course, probably the most important small molecule in all of biology. This is the energy currency of life, okay? And if we look at this sequence a little more, we can see it really consists of 2 parts. There are these black nucleotides on the left and on the right that are base paired in these short stem regions with a loop. And this is really the structural part of the aptamer that kind of holds the aptamer together. And then there's these nucleotides in red, and these nucleotides in red are really the nucleotide sequence that forms the ATP binding pocket. So that's the region of the aptamer that recognizes the small molecule. And we can see these nucleotides up above here in the primary sequence as well. Now, it's really important to point out that these nucleotides are not just any arbitrary nucleotide sequence. In fact, it's a very precise sequence, okay? So if you take any of these and kind of shuffle them around or mutate them in various ways, what's going to happen is you're going to lose the ability to bind ATP. So this sequence is going to lose its functional activity. And so we refer to this, of course, as the sequence structure function paradigm. And this is the mechanism of how aptamers actually function, that you have a sequence, so a primary sequence here, that folds itself into a structure, and it's really the shape of that structure that allows it to have a specific function, okay? And of course, we know this very well for many examples, including this ATP binding example here, because we've solved the 3-dimensional structure of these types of molecules to high resolution. And so again, you've got the 2-dimensional rendering here in the lower right-hand corner of this ATP binding aptamer sequence. And these nucleotides kind of in light green or aqua are these sequences up here that are in light blue, okay? And these are the sequences that once again form the ATP binding pocket. And you can see by just looking at it that this ATP binding pocket is almost perfectly contoured to the shape of the ligand. And then these sequences, other regions of the aptamer, like the ones in white on the left and on the right, that would be these dark blue sequences that again make up kind of the structural domain of it that holds this aptamer together. Okay, so how are aptamers created? Well, they're by and large largely created by molecular evolution. There are a few examples that exist in nature, but the vast majority are created by molecular evolution. So this is a process where you create a library, a random library of sequences that have lots and lots of sequences, And these sequences all fold into different shapes. And some of these shapes will actually bind the target you're interested in. Okay. And so what you do is you take this library and then you query it for sequences that can fold into shape that can bind your target. So in this sort of example here, what we're looking for is sequences that can bind to ATP. And in this case, the ATP is itself tethered to a bead. So if the sequence binds to ATP, it's then retained. These other sequences in red are incapable of folding into the shapes that can bind to ATP. And so they're considered non-binders. And when you take the library and you pass it over beads that are coated with ATP, then these non-binders are lost. They kind of go into the trash can. They essentially go extinct. But these other sequences, like the ones in blue, that can bind to ATP will bind to ATP. And then, of course, you have to recover the binders. So there's a lot of different ways to do that. In the case of ATP, the simplest thing is just to add an excess of free ATP, and essentially the aptamers that are bound to the ATP, which are then bound to the bead, can exchange with the free ATP in solution and just pass through the column, and then you collect them, okay? At this point, you have to then amplify the sequence or make lots of copies of it. So, we typically do that using molecular biology techniques like PCR or reverse transcription PCR. And once you've made lots of copies, you then repeat the process over again. And as you repeat the process, you can adjust the conditions, you can reduce the time, you can reduce the concentration of your ATP ligand, and essentially select for sequences with higher and higher activity. And so over time, you have this kind of survival of the fittest experiment where sequences that were present early on but didn't quite have the activity that could meet the fitness were then sort of lost and go extinct over time. Okay. Now, one thing to keep in mind, of course, is that this entire process is generally considered to be very slow. So historically, what you have is a student. So this is my UCI student here, or maybe a student at another university. And you have your target that you're interested in generating aptamers to. You have your library that you start with. And then, over the course of, say, the next 2 to 3 months, you perform these molecular evolution experiments. Okay. Now, the reason that the process is very slow is that, these functional molecules tend to be very rare, so they're hard to identify. So one way to think about it conceptually is if you think about all of the sequences in your library as a type of sequence space, so the space of your library, and you could perhaps render that in a cone, like the one shown here in orange, where most of the sequences are going to be at the bottom because the cone of the base of the cone is going to be very wide. But there's going to be a small number of sequences kind of near the top, and the ones near the top are going to have greater fitness. So that means they're going to be able to perform the function that you're interested in better than, say, the ones that are below it. Okay. And what happens is that these sequences are so rare that you have to perform, say, 8 or 10 cycles of selection in order to enrich your population enough that you could identify a sequence that you could actually measure its binding activity using, say, an analytical technique like SPR or something. Okay, and so I think it's always good to just sort of conceptualize and think about how difficult this problem is. And one way to do that is just to think about sequence space. So sequence space, I like to render it as just sort of almost like our solar system, where every pixel on this slide is an individual sequence. But as you can see, most of the sequences are dark, meaning they have very little activity. But there are some sequences that are lighting up, and obviously the ones that light up more than others have a greater fitness for the particular function you're interested in. And so just thinking about the numbers, for a typical library that most laboratories would use in the lab, that would be a library that, say, has 40 random nucleotide positions in a row. The total theoretical space would be 4 to the 40th power, because you have 4 different genetic letters, A, C, T, and G, and it's raised to the length of the library. So that's the probability function. And that gives you a number that's like 10 to the 24th, which is a really big number. Okay, so if you do the math, this number is something equal to, say, 20 tons of DNA. So you think of like 20 dump trucks of DNA, so a massive amount. So in the laboratory, we obviously can't make that. So we work on a scale that's still very large, but yet much smaller. And so we're really only sparsely sampling different regions of sequence space. So in this case, a typical experiment might have something like 10 to the 15th sequences, but yet that is still very a large number. So that's 1 quadrillion or 1,000 trillions. Okay. But that's about the number of sequences that we can make and fit inside, say, an Eppendorf tube, but it's still a massive number. So if you wanted to say, oh, well, I'll pause there for a second. So, so the next sort of question that many people are interested in is, okay, they're rare, but how common are aptamers? So what's the probability of identifying an aptamer? And, it tends to be something like, 1 in 10 to the 10th. This experiment's been done a few times, but, they oftentimes tend to sort of focus on a number about 1 in 10 to the 10th. And that number is still very rare. So for example, if you started writing out a 40-mer sequence and you wrote one 40-mer sequence every minute from the day you were born until, say, the day you died, you lived 100 years, you would never come close to this number. So that's just how rare it is and how difficult it would be to find these sequences. So in my lab, what we're doing is trying to identify sequences like this, but that are not composed of DNA and RNA. Instead, they're composed of this artificial genetic polymer TNA. And so we're using, obviously, Darwinian evolution approaches to select and evolve these molecules in the lab. But in the last couple of years, we've also teamed up with the Moonshot Factory at Google. So this is obviously a machine learning group of folks that are very interested in saying, well, this is a difficult problem, but it's a problem that we could potentially either solve or improve or make progress on using machine learning and artificial intelligence. And so what we do is lots and lots of different types of selection experiments for these types of sequences. We take huge amounts of data that's informed of in the form of, say, next-gen sequencing. So oftentimes we're sequencing hundreds of millions of reads per selection. We provide that data to Google. They look at it in their machine learning algorithms, and they send back predictions. We test these predictions and sometimes take those as the basis for, say, another experiment. And so it's a very sort of iterative process that we're doing. And the idea is that if we could do this and we could do it really well, that this could potentially be a strategy for creating therapeutic aptamers. Okay, so these would be aptamers that potentially could compete with, say, human monoclonal antibodies. And the way this would work is you would identify a target that you're interested in. So this would generally be an extracellular target that's involved in a disease pathway. So very common examples would be like cytokines. You would identify that target, you would evolve aptamers that could bind that target, and then you would use the aptamer as a therapeutic to essentially bind the target and inhibit its ability to bind, say, a receptor or another protein in a biological pathway. Okay, so you'd essentially use it as an inhibitor. Now, in addition to trying to accelerate the evolutionary process, we also just overall want to make higher quality aptamers. And so one of kind of the classic problems with aptamers is they tend to have fast off rates. And so any sort of equilibrium binding event like this, where you have a protein, you have your aptamer, when they come together, they're coming together at a certain rate. So we call that the on rate. Okay, and that leads to this sort of bound protein aptamer complex. But then, of course, there's also the off rate, the rate of dissociation, where that complex comes apart and goes in the reverse direction. Okay, and so we can think about that in these sort of plots that are sort of idealized down below. So the problem with most aptamers is they have kind of a fast on rate, but they also have kind of a fast off rate. So think about this like, you're going to a holiday party and you see a relative that you're not particularly keen on. And so, they're coming up, they want to give you a hug, they want to say hi. You want this engagement to occur very quickly, but you want it to end very quickly because you don't particularly like this relative. Versus, say, this plot here on the right, where you see you have a fast on rate and a slow off rate. So this is like the idealized situation where you have this relative that you absolutely adore, you bind to them, but you want this moment to last as long as possible. And so you kind of savor that embrace, okay? And that's what we're looking at, because we're essentially plotting the binding response versus time. So when the binding response is going in the positive direction, you're actually in the process of binding. When it's going in the negative direction, you're in the process of dissociation. So association and dissociation. But again, antibodies have historically done this very, very well, and antibodies not primarily so. So how do you solve this problem? Well, at the end of the day, this is ultimately probably going to be a problem of chemistry. So obviously nucleic acids, whether it's DNA or RNA or TNA or some other XNA, by and large, you have 4 genetic letters. So a chemical diversity of 4. But the problem is these 4 genetic letters are chemically very similar to one another. Okay, you've got amines and you've got carbonyl groups and you've got aromatic rings. Okay, versus say over here on the right, proteins have 20 amino acids with say 7 or 8 different types of chemistries, depending on how you organize them. So they're chemically, it's just much richer chemical diversity. Okay, so one way to kind of get a clue to what sort of chemistry is good for binding is to look at the crystal structures of antibody antigen complexes. And so this is one such structure. So on the left, you've got an antibody. So this is the light chain on top, the heavy chain on the bottom. And then on the right in blue, you have an antigen. And at this interface, you have kind of the paratope surface. So that's the surface on the antibody, and the epitope surface, which is the surface on the antigen. And if you take this complex and you kind of peel it apart and you look at it in what's referred to as the open book view, what you see is on the antibody surface, on this paratope surface of the antibody on the left, where all the residues in yellow are the residues that are sort of making up the paratope. A lot of them have these sort of aromatic ring structures, okay? And this is not a coincidence. So it turns out that if you look at a lot of these different structures, so in this case, it's a sampling of say more than 50 different antibody antigen structures, what you see is that these aromatic amino acid side chains like phenylalanine, tryptophan, and tyrosine are about 5 times more common than say just about any other amino acid residue. And so that means they're really important for binding because they create this large sort of hydrophobic surface that can then bind to the hydrophobic surface of another protein or antigen. And so we decided to go to the trouble of start synthesizing TNA triphosphates that had, a rich set of chemical diversity on them. And we looked at a lot of different groups, not just these aromatic groups, but also different aliphatic groups, as well as cyclic aliphatic systems. Okay, and this was a tremendous amount of work because each one of these triphosphates required 18 synthetic steps. Okay, so that's a long synthetic strategy for producing these molecules. We made here it looks like we're making like 10 different ones. So this is 180 different chemical steps. Now we've worked with all of these, but the ones that we've worked the most with are this sort of phenylalanine and tryptophan analog, where you've got side chains that sort of mimic the phenylalanine and tryptophan amino acids. Okay, and so using these systems, we've evolved aptamers on several different examples of TNA. So TNA aptamers to different targets. This is just one example that we published recently, where it's a TNA aptamer that we now refer to as Threomers because they're functionally enhanced TNA aptamers that have these side chains on them. And they were selected to bind, in this case, it was the S1 protein of SARS-CoV-2. So it's a common target these days. And this is an experimental sequence that we identified in our selection experiment. And what you see is it has this kind of characteristic on rate and off rate that you'd expect from an antibody. You have this kind of fast on rate and then slow off rate. And so the traces like this are very, this trace in particular is very similar to, say, the best antibodies that have been published in the literature against this target. That would be protein antibodies, of course, right? And you see it has a binding affinity in the kind of the high picomolar range, which is where you want it to be for a high affinity interaction, but also one that has a slow off rate as well. So that was, I think, very validating to see that we could generate a very high quality, biologically stable aptamer against an important target. Now, the next thing we wanted to do is, of course, say, well, how well can a machine learning prediction behave in this particular experiment? And so just as a point of reference, this BLI trace shown here in the upper left-hand corner labeled as A, this is again that experimental trace that I just showed you. And so when we did machine learning through our collaboration, they were predicting sequences that would be sequences that would sort of look like things that came out of our library, but would be completely different. So it's a sequence that was most likely almost certainly never sampled in our library. And so this is one example. This is again an example that has the tryptophan chemistry on it. And again, it behaves very, very well. You get a nice fast on rate and a slow off rate and a KD that's even a little bit better than the experimental sequence we had identified. But the one on the right is, again, a computationally predicted sequence based on machine learning. And then I think just to show that this was not like a one-off, we went ahead and did this with the phenylalanine chemistry. And so these are two machine learning predictions based on, again, on experimental data, but they're totally machine learning sequences that now have the phenylalanine side chain that, again, have this kind of fast on rate and slow off rate profile. So I think it was very gratifying to kind of see that. And now we're doing this on other examples as well as we kind of move along. So I think I'll just kind of summarize. We've talked about a lot of different things, but historically, if you look at aptamers and you think about them as sort of a drug discovery funnel, what most aptamer papers do is they start with a very large number of sequences. Generally at least 10 to the 14 sequences. They go through some number of rounds of selection. They get down to a very small subset. Usually it's less than 10 and certainly less than 100 that they then kind of characterize, measure their affinities, and move forward with. And that's kind of been a very traditional approach for, say, the last 30 years. And what we're trying to do is something a little bit different, which is kind of thinking about it as again, like this drug discovery funnel, but really looking at these intermediate populations of sequences and seeing if we can derive information from those populations that allow us to create better solutions. Okay. So I think with that, I'll stop and, obviously thank all of the folks in my lab that are working on this project, of which there are many, the funders that are responsible for supporting the lab financially, and then our collaborators at X as well. So with that, I'm happy to take any questions or, happy to chat about the research work.
Thank you very much, John. That was fascinating. There is a lot of questions in chat. It's kind of traditional for this seminar series that people don't like to unmute themselves. They just ask in chat. But okay, please unmute. If you can read a question before you answer it, that's going to work better for the recording.
Okay, let me
read it out loud, or I can read it.
Yeah. No, I'm just trying to find the first question. Okay, it looks like the first question is from Zach, and it is, has anyone tried to use any XNA polymerase to do oligosynthesis similarly to DNA enzyme synthesis like Twist? Do most people make oligos on templates or chemically still? So I think that's an interesting question. And okay, so there's many parts to this question. So I think the first part is, how do people make, XNA oligos? Doesn't matter if it's TNA or whatever your XNA is. And the vast majority, overwhelming amount of work that has been done is chemical synthesis, making very, very short oligos for, say, hybridization experiments and just traditional kind of nucleic acid chemistry work. So you're making like a 12-mer or 14-mer, and you're measuring hybridization efficiencies with your cell, with itself, as well as, say, cross-pairing with DNA and RNA. There's a very rich literature that goes back many years on that. There's only a small number of XNAs that can be replicated in vitro, just a handful, and TNA is just one of those, using polymerases. And those experiments entirely rely on copying DNA templates into whatever XNA you're interested in, and then XNA back into TNA, okay? And in terms of like moving forward, there's a huge need for improvements in the chemical synthesis of XNAs. So my lab is also working on this problem from just a chemical synthesis point of view, using an automated DNA synthesizer, and trying to make things that would be like a 40-mer or 50-mer that would be entirely XNA, so that we could, for example, validate some of the sequences that we're identifying by our selections or for other reasons as well. But there is a tremendous need also for enzymes that could do programmable DNA synthesis, just like you're seeing at Twist and some of these other companies where they're trying to engineer like terminal transferase or something like that, where you could selectively add one nucleotide and then whatever it is, and then add the next one, and then add the next one. So you could do kind of a more enzymatic approach to solid phase synthesis. And I would say there's been almost nothing in the literature on XNA on that. but it is a rich area and certainly something that, you know, folks should be looking into. So hopefully that captured it all. I know it was a big question. Okay. So let's see. The next one is from Peter. This might be a naive question, but why do you use microfluidic at all instead of just doing selection in E. coli and sorting the bacteria directly? Okay, so bacteria are sort of difficult to sort using like FACS. So fluorescent activated cell sorting is kind of traditional cell sorting approach. It's designed for mammalian cells, which are substantially larger than bacteria cells. And so when you sort bacteria, you know, when you sort mammalian cells, you sort an individual cell because it's so large. But when you sort bacteria, oftentimes what you're doing is you're sorting like a clump of cells, and the enrichment is not particularly good. So, you know, FACS sorting with bacteria has been historically difficult. And so, you know, this is just the way we've developed it. It's once you have it up and running, it's an extremely efficient approach. The downside, of course, is you have to have a lot of, you know, experience with microfluidics to kind of develop these sorts of systems. But I think that sort of technology is becoming more and more available with time. So, you know, that's why we do it. We do it because we want to have a lot of control over the directed evolution of our enzymes, and this just provides maximal control. So in this case, we're using E. coli largely as a factory to produce our enzymes, and then allow them to sort. I mean, there are others that do the same thing we do, but don't, not necessarily for XNA, but don't use E. coli to produce their enzymes. They do it using sort of a cell-free transcription translation system. So hopefully that answered that. Let's see here. David had a question. Are XNA polymerases specific to XNA monomers, or will they take natural monomers too? If the latter, would you speculate that it's possible to make an XNA specific polymerase that can reject endogenous monomers completely? Fascinating question, one we're certainly very interested in. We published a paper, I think, at the end of last year, where we were sort of looking at that. We were looking at kind of crosstalk between the various XNA polymerases that have been created to date, as well as their triphosphates, and saying, okay, you know, how well does our TNA polymerase recognize these other XNA triphosphates, and so on and so forth. The answer is they're all very sloppy. And so I like to refer to these XNA polymerases as generalists. So essentially what my lab and other labs have largely done is introduced mutations that lower the substrate specificity of DNA polymerases and allow them to kind of accept a broader range of sort of modified sugar nucleotides. But I do believe that over time, with enough directed evolution, if it's kind of properly done, that one day we will get to specialists, which would be XNA polymerases that only synthesize XNA. Okay, they don't synthesize DNA. And that's the path that we're working towards. And, you know, we'll see. I think the latest versions that we have are definitely going more in that direction, but it's been a huge amount of work to get there. And, you know, but they all still recognize DNA as well as oftentimes a lot of other XNA triphosphates. However, I will say the scale is tipping because like the one we use, Kod DNA polymerase, the wild type polymerase is a remarkably fast DNA polymerase. It synthesizes DNA at a rate of about 12,000 nucleotides per minute. Now, when you put that into, when you measure those kinetics for DNA synthesis with, say, our third generation TNA polymerase, it makes DNA about as well as it makes TNA, which is at a rate about 10 nucleotides per minute, something like that. So the scale is tipping, and we've dramatically reduced its efficiency for DNA substrates. Yeah. Good question, though. Fantastic question. All right. Stefan has a question. Fantastic talk. Thank you for presenting. When you design libraries for CLEX with XNA, would you use any other design considerations, biases, other constraints that if you design a library for RNA or a DNA afterward? You know, yes, we have played with that a little bit. We've looked at, say, internal stem loops to nucleate a folded structure. We've looked at different sort of compositions and biases and things. But I think for the most part, because a lot of the work we're doing is really sort of methodology development and setting up sort of a platform technology for both directed evolution and machine learning that at least where we're at currently, it's just much easier to have kind of straightforward libraries that have a random region of a certain distance, typically 40, sometimes shorter, and just to have kind of an even distribution of ACTG. So we have looked at those things, but we've largely just kind of returned to this kind of unbiased approach as much as possible. And again, I think it's largely because of what we're doing on the machine learning side of the equation as well. But I think we will go back and look at that some more because I do think biased libraries do have a lot to offer. Yeah. Let's see. Tanner has a question. Would you speculate that there are some constraints that prevent life from sampling some parts of sequence space, both for natural nucleic acids and XNAs? Possibly. I think at the end of the day, sequence space is so incredibly large that the probability of sampling all is just impossible. So I think, my naive view of this is that it's just a statistical question, that sequence space is massive and nature has only sampled a small portion of that. And we see that also in kind of the computationally designed protein world as well, right? Where you can say there's only so many folds that exist in nature. And now there are some groups that are designing, folded proteins that have different shapes than the ones that we've seen already in nature and are natively folded and sometimes even functional. So I think it comes down to a question of sampling. Let's see. Kevin. All right. For such amazingly expanded chemical diversity, do you think we could build a completely synthetic cell based on some XNA system instead of both DNA or RNA, or would you need a completely new ribosome to translate? And if so, is that out of reach right now? Very difficult problem. There are folks in this space that are interested in orthogonal genetic polymers. So what I talked about today are artificial genetic polymers that can base pair with DNA and RNA, and we do it for very obvious reasons that we want to take advantage of all the technology developed for DNA. Now, if you want to create a synthetic cell that is going to have an XNA inside the cell, along with DNA and RNA, you want it to be orthogonal. So you want that XNA polymer not to base pair or interact with any of the natural DNA or RNA inside the cell, because that's really going to sort of mess up the cell. And so there are folks looking at that. And there's an XNA system called PMT, where it's kind of like a derivative TNA, where you've got a phosphomethyl group, and there has been some work on evolving polymerases that will synthesize it. It's a really kind of interesting example because there are at least one example in literature where an engineered polymerase will copy DNA into this sort of pTNA system. But even though the two systems don't base pair. So the polymerase is actually acting as like a molecular clamp that just recognizes the triphosphates and just keeps throwing it in as they go along. And so it's a wild example. We just solved a couple of crystal structures for engineered polymerases with that bound. And hopefully they'll be coming out soon. So it gives us a chance to start to looking at it, from a structural point of view, what's going on the active site. But yeah, I think it's a difficult problem. I don't think it's going to be solved anytime soon. And even if you could do this, how would you translate it with the ribosome? Super interesting questions. I do know that there are folks working on those questions, but I think it's going to take a while. Okay, Anne-Marie, could XNA oligos be used for vaccines? Or they're not translatable like modified sort of mRNA yet. So that's also a very hot area. So there was a company called OmniPossible, which is a synthetic biology company in Europe, and that was one of their goals, is to create the machinery to evolve XNAs or have XNAs that can be translated as a potential strategy for creating new vaccines. And I think they're sort of looking at what combinations are possible for transcription and translation, and could sort of add value as like an mRNA vaccine would, right? So for the mRNA vaccines, a lot of times what you're looking at are pseudouridine nucleotides is the primary, the dominant modification. And it's used to, reduce like an immune response when you receive that as a vaccine. So, perhaps XNAs could function in that way as well. Certainly some of the oligonucleotide therapeutics like ASOs and siRNAs have also taken advantage of modified sugars to kind of control immune responses and things. So, I think it's a totally doable area. It's just a new avenue to start thinking about things. Is machine learning prediction of affinities easier or harder for XNA polymers than natural nucleic acids? I would say it's the same. They're just kind of looking at genetic letters. They're not looking at structures. They're just looking at motifs within kind of sequences and things. So it doesn't matter so much if it's XNA or DNA or RNA. It's just there's so much work involved that it's probably better to do it on XNAs, especially if that XNA eventually has the potential to be a therapeutic, because DNA and RNA are never going to be a therapeutic. They're too easily recognized by nucleases, and they're too easily degraded. So they don't have the pharmacological properties for a drug. So I would say it's about the same. And, it's not my area of expertise, but, based on my knowledge of it. Okay, let's see. Is your information filtering strategy uniquely suited for XNA, or can it be used for other selections too, including kind of protein selections? I'm not entirely sure what you're asking in terms of information filtering strategy. Is that for the machine learning? Possibly?
You can unmute yourself. I would guess she's asking about that, just given by the timing of the question. You were summarizing at the end, showing that you basically go halfway down the pyramid, but I don't know if that's.
I see. No, I mean, I think similar strategies could be done for proteins as well. So, this is like a complex Darwinian evolution system, right? Because we're evolving polymerases, but ultimately we're using the polymerases as tools to evolve XNAs, which we'd like to create for applications. And so, if you want to go down that path, the aptamers or whatever it is you want to make are only going to be as good as the tools you have to make them. And so we're always trying to improve the enzymes. And in theory, the enzymes could be improved using similar types of strategies. Absolutely. So cool. I think that might be all of the questions, but I'm happy to chat about anything else if there's other interest as well.
Thank you so much. I had a couple questions myself, but other people asked it, especially about the fidelity. I'm honestly surprised that the machine learning fidelity is as good as predicting any XNAs, because you'd think it's only as good as the data set you feed it, and you have more data for naturals. So it's nice to know that that's.
Yeah, that's right. So, the data that's available for natural nucleic acid systems, we can't really use for TNA because they're not going to fold the same way or anything like that. But we are producing, I guess there is a lot of data for natural systems. It's not all available, easily available in the literature.
Yeah.
So effectively what we're doing is creating this ourselves. Yeah. So we tend to do lots of selections in parallel and then humongous amounts of sequencing.
I can imagine.
Yeah, it's different. Yeah. Great.
All right. If there are no more questions, I want to say thank you, John, again.
Oh, my pleasure. It was great seeing you all. And again, if anyone has any questions or anything, I'm happy to follow up via email.
Thanks again, and bye, everyone.
Okay. Bye now.